Hello and welcome everyone to this video podcast for Human Genetics. In this podcast, we're going to talk about one of my favorite topics in the whole book, and that is gene regulation. As I was walking to work today, I was going over the notes I had, reminding me what I wanted to say in this video podcast. And I usually open this up talking about some famous symphony, like Tchaikovsky's 1812 Overture. And as I was walking in, Queen's We Will Rock You came up on my playlist. And as I was listening to this song, I realized that that song is such a better song to explain this with. You'll have to use your imagination here as we go through this, but I think it's a pretty good way to think about gene regulation. So before we begin with the rest of the information of this podcast, I want to take 2 minutes and listen to this iconic song. So just sit back and listen to the song, and then I'll come back and explain to you why I think it's a pretty good analogy for gene regulation.
Buddy, you're a boy, make a big noise, playing in the street, gonna be a big man someday. You got mud on your face, you big disgrace, kicking your can all over the place, singing, we will rock you. We will rock you. Buddy, you're a young man, hard man, shouting in the street, gonna take on the world someday. You got blood on your face, a big disgrace, waving your banner all over the place. We will rock you. Sing it. We will rock you. Buddy, you're an old man, poor man, bleeding with your eyes, gonna make you zombie someday. You got mud on your face, big disgrace, somebody better put your bag into your place. We will rock you. Sing it. We will rock you. Everybody, we will rock you. We will rock you.
All right, welcome back. Hope you enjoyed the song, and now you're probably super pumped to learn about gene regulation. But let's think about this analogy that I was thinking of on my way into work. So we have this song, We Will Rock You. And as you know, when you listen to this song, there is this constant beat throughout the whole 2 minutes of the song. It's a short song. Throughout the whole song, you hear. You might like to know that of all my musical skills, that is the only one I have. And it's not very good, but it's all I got, so I'll do it frequently. So throughout the whole song, you have this constant beat. And then, about 12 seconds into the song, 12 seconds in, you hear the singing. This constant beat is a very iconic, you hear that and you know exactly what song's coming. But when the lyrics come in, it makes that song a little bit more. It adds to that song. And until that, now for the most of the song, that's what you hear, those amazing lyrics plus that constant beat. At about 1 minute and 20 seconds, you hear some electric guitar. It starts off slow and then it ramps up and then it takes over the song. But even still then, you still hear this constant beat. Then about 2 minutes, it ends abruptly. Almost sad. You kind of want more, but 2 minutes in, it's over. This song here is characterized by specific lyrics, instruments, notes, etc. to make it unique. Now, what about a cell? How is this similar to gene regulation in a cell? And for this example, let's just think about pancreatic. We know that in a typical pancreatic cell, there's going to be these housekeeping genes. When I say housekeeping gene, what I mean are those genes that are always on. They're always on and they're necessary to maintain the cell as it is. They're important in things like energy, and I'll abbreviate energy like so, and other homeostatic functions, like maintaining its shape, maintaining its pH, maintaining its temperature. All of these things are housekeeping genes. And while they're important, they're similar to what you might see in other cells. So while hearing this is very iconic, and when you hear it, you know you're going to start hearing We Will Rock You. In itself, it's not that masterful, right? That could be in any song almost. It's not, but it could be. There's nothing really sophisticated about that. I'm not trying to downgrade the awesomeness of that song. Now, in order for this cell to be a pancreatic cell, and say not a heart cell, is specific genes have to be turned on. I'm going to say activated. If it's a pancreatic cell, certainly insulin genes have to be turned on. And then let's just say heart genes, any genes necessary for heart function, have to be turned off. Just like we see in 12 Seconds, where you start hearing those lyrics in the song, that makes that song uniquely We Will Rock You. Just like turning on insulin genes makes this pancreatic cell uniquely a pancreatic cell. Okay, so these genes now, this on and off of certain genes make it uniquely pancreatic. Having this very specific singing that comes in at 12 seconds makes this song We Will Rock You. Now, at about 1 minute 20 seconds, I said, the electric guitar takes over and it kind of starts off slow, goes really fast. We know that in cells, and in pancreatic cells are no different, that while these specific genes have some on and off activity with them, we have these housekeeping genes, every now and then other genes are activated in times of stress. And for that period of time, they sort of take over. And that's what happens at 1 minute 20 seconds. The electric guitar comes in, takes over the song. Still, We Will Rock You, but it's changing. Same thing with some of these stress-specific genes that have to be turned on at certain times. Now, sadly, just like this song ends, cells die. Not quite as abruptly as this song does. The cells enter into a cell death pathway that is very orchestrated, and it's not quick, but not every analogy is perfect. Now, I'm going to come back to here that the specific lyrics, the instruments, the tunes, the notes that we heard in We Will Rock You are unique to this song, as we've said. When you're listening to the song, you don't hear, let's say, this other song at the same time. All right, I won't make you listen to all of that, but it brings me back to my good old days. But when you hear one song, you don't hear the other one, because different instruments are used, different lyrics are used, different notes are used, different tempos are used. Each song is unique, just as if we were looking at a heart cell. The things that make these songs unique, different genes, are expressed in the heart cell to make it unique. It's going to have its own set of housekeeping genes. It's going to have specific genes that give it heart function. And there's going to be other genes that kind of take over at certain times of stress. And it too, at some point, will die. But it's unique because of these specific genes that are turned on. I think that's all I want to say about this analogy. I hope it made sense and I hope it helped. But now what I'd like to do is I'd like to bring it back to more of a biological explanation beyond just what we saw here. Okay, so I'm going to talk about gene regulation in a slightly different way right now. And I'm going to go back to a topic that when I first learned about it as a college student, totally geeked me about genetics and cell biology, and I've never looked back since. And that is, if we can remember, way back when we were a zygote, a long time ago. It's back in 1969 for me, but these were good times for me. At the time of being a zygote, I had approximately 23,000 genes. I may not have known it, but life was pretty good as a zygote. Now, we talked a few chapters ago how this zygote goes through many cell divisions and many stages of development to ultimately become a fully formed being. Now this next part is the part that cannot be overemphasized enough because it's super important. As this adult, say me, I have about 30 trillion cells. And that in itself is pretty amazing because I went from one cell and through the process of mitosis, I eventually have now 30 trillion cells. But the part that is amazing to me is that in each of these 30 trillion cells, in each of them, I have 23,000 genes. The same 23,000 genes I had as a happy zygote. Throughout all of these mitotic divisions, I've maintained these same genes. And for the most part, without any mutations. Now, there are some errors. We'll talk about mutations in the next chapter. There are some errors, but for the most part, this is an extremely faithful process of producing all of these cells with the exact same number of genes. The genes in my big toe cells and the gene in my heart cells are the same genes that I had in 1969 when I was a wee little zygote. Even though the cells in my big toe and the cells in my heart have the same genes, we know that these genes are what we say differentially expressed. We don't want my heart to express genes that are necessary for big toe functions, whatever big toe functions are. We don't want that in my heart. So to talk about how we differentially express genes, I'm going to use hemoglobin as an example. Humans have many hemoglobin genes, and we're going to talk about these 5. And they're called alpha, beta, epsilon, gamma, and zeta. We know that these genes will be expressed at different times, and ultimately what will happen is they will form a 4-protein molecule called hemoglobin. Each of these 4 subunits will be expressed by one of these genes. They'll make this hemoglobin. Now, hemoglobin has a few functions, but the 2 I want to focus on is delivery of oxygen and removal of CO2. CO2 in our body is toxic, so we want to get rid of it. Oxygen in our body is needed for cellular respiration. So these 2 things are both very important, and hemoglobin does that. Now, through the course of evolution, turning the correct genes on at the correct time has proven advantageous. For instance, we know as an embryo, those first 8 weeks of development, that our embryonic hemoglobin has 2 subunits of epsilon and 2 subunits of zeta. And so what happens at the gene level is epsilon is turned on and zeta is turned on. When I talk about gene expression and I show a gene with an arrow like this, that means it's turned on. Now in the embryo, with those being turned on, alpha, beta, and gamma are turned off. That's my symbol for turning it off. Just a straight line there showing you that it's being blocked. Now, as that embryo further develops and say becomes a fetus and makes fetal hemoglobin, a different subset of genes are turned on. But first, we're going to make sure we turn off zeta, and we're going to turn off epsilon, and we're going to keep beta off as well. But now, as a fetus, we're going to express alpha and gamma. So fetal hemoglobin will look like this: 2 alphas and 2 gammas. Prior to birth, a different form of hemoglobin is turned on. We call it adult hemoglobin, but it's starting to be made as the baby is born. So adult hemoglobin will still make these 4 subunits, but now zeta is still off, gamma is now off, epsilon remains off, alpha is still expressed like it was as a fetus, but now we get beta expressed. So we have 2 alphas and 2 betas as the adult form. As a biologist, when we see an interesting phenomenon like this, where different genes are expressed to produce different proteins at different times of development, and we see this in humans and we don't see it in other animals, the question is, at the evolutionary level, is why? Or what advantage was provided? And we should think about 2 things here. And the first thing we should think about is the environment. We know that oxygen abundance, or concentration I could say, increases from embryo to fetus to adult. As an embryo, there's very low oxygen. As an adult, relatively speaking, we have oxygen all around us. It's a pretty high concentration of oxygen compared to the embryo or fetus. The fetus to oxygen abundance is, let's signify that with a symbol like this, an up and a down. It's somewhere in between. The other point we should consider is this, and that is oxygen affinity. Turns out in lower oxygen environments, we have higher affinity. Or I should say, the embryonic hemoglobin has higher affinity. By affinity, I just mean the ability to bind oxygen. These proteins bind oxygen with greater affinity than the adult hemoglobin. So here, adult hemoglobin has a lower affinity. It's important to remember that just because there's a lot of oxygen around, we don't necessarily want it to bind super tight, because we want it to bind enough oxygen that's needed and have an easy way of releasing it into the tissues once it gets there. If it binds it too tightly, it can't release the oxygen as efficiently. So it's this balance between the oxygen environment, the concentration of oxygen, and the affinity of the different hemoglobins that provides that evolutionary advantage throughout human development. One thing we hypothesize about with human development is that this change in affinity for oxygen, depending upon its abundance in the environment for the different stages, allows for oxygen to be delivered to developing tissues or organs at a more efficient level. We believe this has greatly enhanced our brain development as a fetus, and ultimately that has led to advantages, one would think, as an adult. Now the next question I have is, do we care? Well, as a biologist and as a geneticist, I care because I think this is really amazing, but I'm a bit of a nerd. And so as I try to explain this, say, to my parents or some of my friends who aren't scientists, and they say, well, why do we care if it does that? It's neat, it's helpful, but does it really benefit me? Yes. Often, I'm going to write this down here, basic research leads to clinical discoveries. What we learn about understanding hemoglobin expression has led to some clinical discoveries that I'll talk about in a moment. I should also say, when I say basic research, what I'm talking about is that general question of asking, why does something happen? And often scientists, when they are studying basic research, she may or she may not even think about this clinical aspect of it until later. She's doing this research because she cares about the general phenomena of life. Later, we sometimes find clinical applications of this basic research that she may have discovered. And so I want to talk about sickle cell anemia. We're not going to spend a lot of time with it right now, but you should know that sickle cell anemia occurs when there is a mutation on the beta gene. One nucleotide mutation. So it's a very small mutation, but it causes the blood cell to go from being very flexible and compact to being sickle-shaped. This sickle shape can cause pain and it can cause death. So if there was some way we could go from sickle shaped to regular shaped, that could decrease pain and hopefully increase the life expectancy of somebody with sickle cell anemia. So how does this help us? Now remember, we have a mutation in the beta gene, and that leads to this sickle cell shaped red blood cell. Beta is not expressed as an embryo, it's not expressed as a fetus. So they hypothesized that if you could take someone that was sickle cell, so let's write a sickle cell patient as SCA, and you convert alpha beta hemoglobin, this hemoglobin here, to the fetal hemoglobin, alpha gamma here, could that effectively treat sickle cell anemia, preventing pain, preventing death? And what they discovered was that yes, if we could find a way to allow someone with sickle cell anemia as an adult to express alpha and gamma, so that would be like this, and now shut down the expression of beta, that indeed when we do this, the people no longer have sickle cell anemia. How do we do that? We won't go into great detail here, but we know if we give someone with sickle cell anemia, remember with that alpha beta hemoglobin, and we give them hydroxyurea, that they will now express the alpha and gamma forms of hemoglobin. Red blood cells now are not sickled, and the patient is healthy. You may wonder if they can be as healthy since they're expressing a fetal hemoglobin, and it's not perfect, but they don't have sickle cell anemia, and they survive fine, and they have no huge consequences to switching to fetal hemoglobin. And we know this because a researcher mapped out the expression of these genes, figured out how we could change those expressions of those genes, and now we have a very effective treatment for someone with sickle cell anemia. I would like to now begin with this question. Why is it that we are learning so much more about gene regulation today than we were, say, 30 years ago or 20 years ago? Well, back when I was in graduate school, for the most part, we studied one gene at a time. We would see when it was expressed, we would see what happened when we mutated it. Sometimes we would bring in another gene, so we might study one or two at a time. And we were learning a lot of really great things, and it was a lot of fun, but ultimately it goes kind of slow that way because you don't want to just learn a few words in a song. You want to understand how all those words come together to generate the whole song. And studying one gene at a time wasn't really very effective at that. And so that initially led to studying genomics. And genomics is the study of structure, function, and evolution of the genome. And this is effective and useful, But the genomics of any given cell is, for the most part, the same. It's not when we start moving into the field of transcriptomics that we begin to learn a little bit more about specific cells. With transcriptomics, we look at all the messenger RNA in a specific cell or tissue at a given time or condition. So with this approach, you can look at the messenger RNAs in your heart cell versus your pancreatic cell. And by looking at the messenger RNA, you know more than just which genes are present. You know which genes are making messenger RNAs. And so that tells you that that gene that's making messenger RNA in a pancreas cell and not in a heart cell, then that gene is more specific to the function of the pancreas. So we got a lot of really useful information with that. We also moved into this other area called proteomics. And that's essentially the same here, except we're looking at proteins. You get very similar data that you do with transcriptomics, except looking at the proteins doesn't just tell you that the messenger RNA is present. It says that that messenger RNA was produced and ultimately it made this protein. So it's a little stronger evidence of a gene functioning in a tissue than just transcriptomics. Now, if you look in your book or you look anywhere on the internet, you'll see there's a whole bunch of these kind of omics. It's a very popular thing to do is put omics in front of a particular broad area you're studying. And all of those are really important, but these are the three I want you to know for this class. So we know that different cell types will express different genes. We know that different cells as an embryo versus an adult will express different genes. But the big question is, how do our cells know which genes to turn on or off? Well, I hate to end this podcast so abruptly, but it seems like a convenient place to stop. In this first podcast, I introduced to you some of the important terms and concepts associated with gene regulation, but now we're ready to step into a slightly different direction. And we're going to start talking about the mechanisms that help control which genes are regulated and which genes are not regulated. So if you want to know the answer to that question, you're going to have to watch the next podcast. So hopefully I'll see you there. Bye.